Hello everyone and welcome back to our channel. If you haven't hit subscribe yet, please do. Today we'll be discussing drug metabolism. Drug metabolism will be divided into three parts by myself. We prepared and we're going to be presenting the slides for this chapter by referring to Foy's Principle of Medicinal Chemistry, the 8th edition. In short, we're going to be starting off with an opening scenario and then move on to the lecture material and provide few examples to help you better understand the material. YH, a 32-year-old female with a history of chronic schizophrenia, presented for a routine follow-up visit to the behavioral health clinic. Her psychotic symptoms had been well controlled on risperidone 3 mg daily. At her previous appointment a month ago, YH had reported being depressed, so her psychiatrist initiated a trial of paroxetine 20 mg daily as treatment. As the pharmacist was interviewing YH for medication history, he noticed that there was a resting tremor in her left hand. YH acknowledged that it had first appeared within the last few weeks. She also stated that she was just starting to feel less depressed. The reference of this case was taken from the Foy's Medicinal Principle book. To introduce what drug metabolism is, you have to know that first of all, drugs were first developed and prescribed under the old paradigm that one dose fits all, which largely ignored the fact that human beings are genetically and metabolically different, resulting in variable responses to drugs and even drugs within the same dose. Drugs can no longer be regarded as chemically stable entities which elicit a certain desired pharmacological response, after which they're excreted from the body. Basically, you have to know that drugs undergo a variety of biochemical changes brought about by different enzymes found in different locations within the body, with subsequent alterations in the nature of their pharmacological activities, duration of activity, and toxicities. And from this aforementioned point comes the definition of biotransformation or drug metabolism. It's the biochemical changes of drugs and other xenobiotics which can help in their elimination from the body. Elimination is not the only purpose, and you have to know that xenobiotics are foreign substances which enter and become part of our biological systems. As can be seen here, we can notice that metabolism can either lead to inactive, less active, or more active products. Drug metabolism allows the body to terminate the action of some drugs by transforming them into pharmacologically inactive products. However, some inactive drugs, called prodrugs, require biotransformation to become pharmacologically active drugs and exert biological activity. Traditionally, metabolism was always linked to the termination of drug action and system detoxification. However, this is not accurate because some reactions can produce toxic products, like as can be seen in the below, in order to form carcinogenic or mutagenic compounds which create side effects and cause damage, as they can bind sometimes to unwanted receptors. In the following series of slides, we're going to be giving examples of metabolic reactions. First of all, biotransformation can produce pharmacologically inactive molecules which are readily excreted. This is the example of hydrolysis of procaine or procainamide by esterases and amidases, respectively. Another significant example would be biotransformation into pharmacologically less active molecules. For instance, take the example of cytochrome P450 oxidative and dealkylation of propranolol to norpropranolol, or the example of N-dealkylation by CYP450 of morphine to normorphine. A third example would be biotransformation into pharmacologically more active molecules. For instance, cytochrome P450 mediated oxidative O-dealkylation of codeine into morphine, which is a more active metabolite. This will also be discussed in part 2. We can also take the example of esterase hydrolysis of diamorphine into morphine, which is a more active molecule. Another important biotransformation reaction would be the biochemical changes that can activate an inactive prodrug and form an active drug. Take the example of hydrocortisone succinate sodium. It's a prodrug. However, after it's been taken into the body and hydrolyzed by plasma esterases found in the blood, it's going to be transformed into hydrocortisone, or the active component. It's a steroid hormone. Another example would be the anti-cancer drug irinotecan. It's basically a prodrug. However, after it's been hydrolyzed by hepatic carboxyesterases, it can form an active moiety. Hydrocortisone and hydroxycompotecan, both drugs have low aqueous solubility, and they're going to be linked to hydrophilic groups, or in order to form sodium salts, for instance, like in the case of hydrocortisone succinate sodium, in order to make these compounds more water soluble and produced in the form of IV maybe, in order to be suitable for injection. Lastly, biotransformation may generate a pharmacologically toxic metabolite. Thalidomide, an anti-nausea and a sedative drug which was introduced in the late 1950s, was used as a sleeping pill and quickly discovered to help pregnant women with the effects of morning sickness. So it was an OTC drug used for preventing morning sickness in women. It was sold until 1962, when it was withdrawn after being found to be teratogenic and causing several forms of birth defects. More than 20,000 children in different countries were born with deformities such as phocomelia as a consequence of thalidomide use. This is because of the racemization of R-thalidomide to S-thalidomide by isomerases, having S-thalidomide as the toxic product. In the slide, you can see more examples which can help you understand the importance of metabolism in different aspects. First of all, it could be to detoxify the body from a certain compound. We could also have the formation of products with similar activity compared to the main original drug. You can also have the formation of products which have different activity than the parent drug. So we can have the formation of isoniazid, an anti-tuberculosis drug, from iproniazid, which is an antidepressant. We can also have, sadly, the formation of toxic metabolites, which can cause harm to the body, such as the transformation of phenacetin into anhydroxyphenacetin, which is hepatotoxic. Now, after we've discussed examples and what drug metabolism is, we have to know its importance when talking about real-life examples. Let's take the case of lipophilic drugs, which are not completely ionized at physiological pH. If their drug elimination or termination of action only relies on drug renal excretion, such drugs won't be eliminated, sadly, and would have to be filtered back by tubular reabsorption and thus circulate in the body. And this could be for long periods of time, and thus promoting extremely prolonged duration of action, or DOA. Thus, there definitely has to be another route which can help transform such lipophilic molecules via a series of stepwise biochemical reactions into more ionizable and readily excretable water-soluble products. For instance, I can give you the example of people that have renal dysfunction, so they have problems in their kidneys. They have to take drugs mainly that are metabolized maybe by the liver and not by renal excretion. This is one example of tailoring therapy by physicians in order to make sure that proper ADME process is taken into consideration: absorption, distribution, metabolism, and excretion. Now, we've discussed lipophilic molecules. What about small molecular volume size polar molecules? These molecules can undergo renal excretion, which is a process that plays a very important role in their elimination. You have to know that we have various sites of drug metabolism. The liver is the primary site. It's highly perfused and highly rich in enzymes which act on endogenous and exogenous compounds. A very important process also, which will be discussed in a few minutes, would be the FPE, or the first pass effect. It also occurs at the level of the liver. Extrahepatic metabolism sites, which you might heard of, would be the intestinal mucosa, the plasma or the blood, the kidneys, the brain. This might be unusual to some. The lungs, the adrenal glands, the placenta, and the bacterial flora. Diseases of the liver can affect the metabolism of drugs which mainly undergo hepatic elimination. However, if we have renal disease, but the drug is metabolized, for instance, at the level of the brain, then the drug metabolism of the drug in question will not going to be affected by our condition. As promised, we're going to explain the first pass effect in details. We have two pictures to the right which are going to elaborate on what I'm going to be talking about. The ability of the liver tissues to metabolize substances into either pharmacologically inactive or bioactive molecules before they reach the systemic blood circulation is going to be called the FPE, or the first pass effect. The fraction F of an orally administered drug which successfully passes through the gut lumen and gut wall is then taken via the hepatic portal vein, which you can see in the picture at the bottom, where metabolism of drugs by enzymes takes place. The extraction by the liver of orally administered drugs is called FPE. So as a conclusion, take a solid dosage form or an orally administered drug. This tablet would get dissolved in solution, then the drug particles will be absorbed from the GI tract, gastrointestinal tract. The drug particles will then reach the liver, undergo FPE or first pass metabolism, and then reach the systemic circulation before distribution to the site of action and then elimination. An important thing would be to note that not all dosage forms undergo first pass effect. We might have certain dosage forms that can bypass the first pass effect, and this is needed in some cases. This could be the sublingual route or the IV route, having the IV route directly enter the blood circulation and have access immediately to the rest of the body. However, as we said, orally administered drugs are first exposed in most cases to the liver and might be extensively metabolized before having only a small percentage reaching the rest of the body. Now, drug bioavailability is a term you're going to be hearing a lot in your future professional years. It's basically the fraction absorbed into the systemic circulation. Take the example of an orally swallowed drug. The parts after the solution will enter the digestive system and then travel via the hepatic portal system to reach the liver, whereby drug metabolism is going to occur, the first pass effect we've discussed, and then we're going to have distribution to the rest of the body and to the target site. The greater the first pass effect, the less the agent generally would be able to reach the systemic circulation when taken orally, and thus we're going to have lower bioavailability and possibility for decreased therapeutic response. There's also, of course, a formula for bioavailability, which you're going to be taking in your future kinetic courses, which are going to relate the PO versus IV and the area under the curve in a mathematical way. Just for your information, here's a list of some drugs which undergo extensive first-pass effects, such as nitroglycerin, propranolol, sympathetic catecholamines, morphine, clonazepam, hydrocortisone, testosterone. You can see that they're from different pharmacological classes which have very different activities. And we also have a list of drugs which undergo little to no first-pass effect, like sulfamethoxazole, metronidazole, tolbutamide, or pendolol. Take the example of lidocaine IV versus PO. We know that lidocaine is an antiarrhythmic. We know that despite being still available orally, the oral route of administration is not preferred. And this is because of lidocaine's extensive metabolism via first pass effect into inactive metabolite. So it's almost ineffective orally. Thus, unless recommended by physician otherwise, it's mainly used IV due to extensive first pass effect into inactive metabolites. However, as we've said, the oral dosage form is still available. Another example would be nitroglycerin, which is very liposoluble and readily transported to membranes. Therefore, it's totally absorbed by the intestinal mucosa. However, it undergoes a massive FPE, rendering its oral bioavailability almost negligible. So as you can see in the picture to the right, it can be given in different routes. So we can give it sublingually, buccally, transdermally in order to avoid first pass effect and bypass this. If we want to translate the first pass effect into the research setting, we can conclude that FPE can be a disadvantage for some drugs but an advantage for others. In scientific pharmacokinetic research, testing is done. For instance, you've administered an orally available drug and you've seen that we have extensive first pass effect into active and inactive metabolites. Take the example of a drug X that has been extensively undergone first pass effect after high absorption from the GI tract. Consider that the majority of this drug is going to be metabolized into inactive metabolites as the major end product. What can you do now as a solution? Would you keep the market filled with this drug knowing that it's mostly going to be metabolized into inactive products? As a solution, researchers have thought of other ways or other routes to administer the same drug, such as the sublingual route, the intravenous route, the subcutaneous, intramuscular, the topical, or even the rectal route, depending on the pharmacokinetic and the pharmacodynamic properties allowing for such changes. Factors affecting drug metabolism. We're going to be giving you examples to help you better understand this concept. First of all, one important factor is pharmacogenomic changes or genetic variability. This means that we have certain populations within the global scale that have genetic polymorphisms or mutations in enzymes which metabolize a certain drug, and this makes them classify into certain categories. So for instance, rapid or poor metabolizers. Now, when it comes to changes in age and gender, we have a lot of examples that can help you understand this concept. Many biotransformation reactions are slowed in young and elderly people who have decreased drug metabolism capacity due to having less CYP enzymes, having less liver mass, and less blood flow to the liver. And thus, they have to be prescribed lower drug doses in order to minimize possible drug reaction toxicities. Another concept is the fact that the enzyme system at birth, especially in a preterm baby, is functionally immature, and especially for oxidation and glucuronidation reactions, which are also going to be elaborated further in part 2. Neonates also carry out most, but not all, of the oxidation reactions, as the enzyme systems are becoming mature only after the first 2 weeks of life and continue to be so after childhood. Neonates also have a decreased conjugating ability. Thus, jaundice is one of the results due to deficient bilirubin conjugation. Also, you can also have chloramphenicol causing gray baby syndrome, and this is due to decreased conjugation capability. Now, in elderly, drugs like antidepressants known as TCA family or tricyclic antidepressants and antiarrhythmics persist for a longer time in elderly and in higher concentrations. So, morely, we're going to have more central nervous system toxic side effects. Moreover, dietary changes can affect drug metabolism. Take the example of grapefruit juice, which can inhibit CYP3A4 activity. Take the example of smoking, which might induce CYP1A2 activity, or even char-grilled foods. Another example would be discussing how environmental changes can affect drug metabolism. Industrial workers get exposed to certain pesticides, which might be CYP inducers, and thus cause faster drug metabolism, which might be undesired in certain cases. Another example would be, for instance, some people may have a faster drug metabolism and thus feel the effect of a local anesthetic kick in more rapidly than others would. Back to diet. Poor diet and deficiency in lipids, proteins, vitamin C, vitamin B2, and vitamin E can decrease enzymatic capacity to metabolize various drugs. Now, if we're talking about the importance of pharmacokinetic and pharmacodynamic changes, you have to know that dose, frequency, and route of administration are very important. Enzyme saturation might affect the drug response depending on the drug dose. Also, we might have some drugs stored in adipose tissue because they might be more liposoluble, and thus they might not be reaching the liver and thus metabolized into their active entities. You can also have plasma protein binding hinder the effect of certain drugs and decrease drug metabolism, since the enzyme might be more bound and thus less free and thus less able to reach the receptors. Finally, we are going to conclude with the importance of disease-drug interaction. Liver diseases might be a very important topic to discuss because we know that some people might have hepatitis, cirrhosis, liver cancers, fatty liver, and other acute inflammatory diseases. And thus, this can slow down or sometimes impact the metabolism of certain drugs. For instance, we know that hepatocytes, which are the cells of the liver, and a very important fact is the importance of blood flow to the liver. This can cause an increased systemic availability of such drugs like labetalol, propranolol, which we know that they have a fast and high first pass effect. So, if we have acute inflammatory liver diseases, we have more systemic availability of such drugs, and thus more prolonged half-life and decreased clearance. Also, cardiac diseases can slow down blood flow to the liver and decrease liver metabolism. Some thyroid diseases, such as hypothyroidism, can decrease drug metabolism. On the contrary, hyperthyroidism can accelerate drug metabolism. Lastly, the stereochemistry of drug molecules can affect the way they're going to bind to enzymes and drug receptors. You have to know that a drug might interact with food, herbs, genes, allergies, diseases, and most importantly, with other drugs when taken concomitantly. These might be classified and categorized as desirable or non-desirable, and you can refer to lexicon in order to analyze the drug-drug interactions that do exist and categorize them into A, B, C, D, and to see if you have to change the drug therapy, the drug regimen, the route of administration, and other possibly important pharmacokinetic entities. Some relevant drug-herb interactions would be understanding what St. John's wort is. It's basically used as an antidepressant, and also it's categorized as a CYP3A4 inducer. So you shouldn't be able to take St. John's wort with drugs that are going to be metabolized by CYP3A4 enzyme, because this drug in question would be highly metabolized more and more, and thus this would be causing unwanted and undesired effects on the body. Echinacea, used for cold and viral infections, inhibits hepatic CYP1A2 and intestinal CYP3A. However, on the other hand, it induces hepatic CYP3A activity. So, echinacea should not be given by a drug that's metabolized either by CYP3A or CYP1A2. Ginkgo biloba, which is used mainly to improve memory, induces CYP2C19 activity. And lastly, kava, used to treat anxiety and insomnia, is a potent inhibitor of many CYP450 enzymes. Now, why did I include these examples? Because you have to know them as pharmacists and as pharmacy students, and make sure that you also study drug-herb interactions when you're taking the patient case and when you're doing a best possible medication history exam, because these also impact the pharmaceutical care process and the patient outcome. As previously mentioned, for drug-drug interactions, we're going to be giving you examples, and we also said that you can refer to Lexicomp in order to place the list of the drugs the patient is taking and click on Generate in order to have a full detailed report of the possible drug-drug interactions that are going on in order for you as a clinician to change the therapy, modify it, change the doses, change the medicines, and optimize patient care. This slide provides you with a list of drugs which can enhance or inhibit drug metabolism in humans. But for the sake of this presentation, we're just going to tackle few important examples which you might experience and you might come in hand with in your experiential courses. Not all DDIs are bad and should be avoided. Some ones are desired and can be classified as beneficial. You can take the example of cyclosporin and CYP3A4 inhibitors. Cyclosporin, an immunosuppressive agent, it's used basically in organ transplantation to decrease the immune response and thus not to reject the transplant. It's very expensive and metabolized by CYP3A4 into inactive. If co-administered with a CYP3A inhibitor, this allows reduction in dose and cost because would be inhibiting the inactivation caused by liver metabolism. In other words, cyclosporin metabolized by CYP3A4 into inactive is a very important and very common mechanism. If we give a CYP3A4 inhibitor, would be inhibiting the metabolism into inactive compounds and thus would be able to give less dose and decrease the cost burden on patients. Another example would be undesired drug-drug interactions like grapefruit juice and statins causing rhabdomyolysis. Also, grapefruit juice, rich in minerals and vitamins, inhibits intestinal CYP3A4. So, we might have increased bioavailability of drugs metabolized primarily by intestinal CYP3A4, example diazepam, methadone, carbamazepine, or nifedipine. And thus, the metabolism of these drugs is inhibited by grapefruit juice, so you shouldn't be taking these drugs concomitantly because you might have changes in plasma levels and cause toxicity to the patient. Based on genetic polymorphisms, which we've discussed as a very important factor which impacts drug metabolism, you can take the example highlighted in red of nortriptyline, which is a TCA antidepressant. We have polymorphisms of the CYP2D6 enzyme, which is a very important enzyme that metabolizes nortriptyline. You have different doses per day, and on the right you can see that we have different phenotypes: ultra-rapid metabolizers, URMs, extensive metabolizers, EMs, intermediate metabolizers, IMs, and poor metabolizers, PMs. Take the example of this antidepressant nortriptyline metabolized by CYP2D6. For instance, in poor metabolizers, we have decreased clearance of the drug, and this might cause various effects of overdose like serotonin syndrome, arrhythmias, cardiotoxicity, and sedation. Another notable example would be the metabolism of S-warfarin by CYP2C9. In poor metabolizers, we might have decreased drug clearance and more risks of bleed. If we have another example, the extensive metabolizers, which we can see that it takes part as 80 to 65% as a frequency in Caucasians. We need to increase the dose of the drug as it would be highly metabolized into inactive and thus ineffective at lower doses. Glycoprotein is a cell membrane-associated protein which transports a variety of drug substrates across the cell membrane, and this can help in their excretion. To the right, you have a list of different examples. Take the example of digoxin, used for congestive heart failure. Its elimination into the intestine and urine is mainly mediated via P-glycoprotein. Grapefruit juice inhibits PGP. Also, quinidine inhibits PGP. So, there exists a drug-drug interaction when concomitantly administering to the patient grapefruit juice or quinidine with digoxin. This is an undesirable interaction, and we're going to explain it. Co-administration of quinidine or grapefruit juice, which also act as PGP inhibitors, with digoxin causes an undesirable DDI. Quinidine or grapefruit juice inhibit digoxin elimination, and thus we have digoxin drug accumulation and possible toxicity, cardiac toxicity, visual, neurological, and even digestive. But we know that digoxin is a narrow therapeutic index, an NTI, and any slight changes in plasma levels can cause toxicity, so we shouldn't co-administer digoxin with quinidine and grapefruit juice. This illustration helps you classify drug metabolism reactions into two distinct categories, being phase 1 or phase 2. These are two terms that are going to follow you during the drug metabolism explanation that I'm going to be continuing in part 2 and part 3. Basically, this categorization is not based on the site of action, which is the liver. It's the same, but it's based on the type of metabolic chemical reaction the drug is subject to. Now, why would we categorize and why would we study drug metabolism? Familiarity with the mechanisms of drug metabolism can often aid you predict consequences of drug-drug interactions, drug-food, drug-herb interactions, and help explain patient adverse responses to drug regimens. Such knowledge can help assure improved selection of proper drug regimens and doses before therapy begins. And in this chapter, you're going to be helped and explained with different metabolic reactions and different drug products, which are going to be classified as active, inactive, possibly toxic, major, and minor drug products. If the drug is polar enough, part of it will be directly excreted. However, if it already contains a polar functional group, it can be directly conjugated, meaning you can go with the next arrow to the left, directly move from drug to phase 2. If the drug after phase 1 reaction becomes polar enough, it will be directly excreted. Otherwise, the drug needs to be functionalized, conjugated, and then excreted. This is a very important slide because it shows you the names and the classifications of the phase 1 and phase 2 reactions. Phase 1 are classified into oxidation, reduction, and hydrolysis, having oxidation as the majority. Phase 2 are classified depending on what group you're going to be adding onto the parent drug. Phase 1 reactions are termed as non-synthetic functionalization reactions. Non-synthetic means that the same molecule is oxidized or reduced. In phase 1, we're introducing or unmasking a polar function group, example an OH, a COOH, an NH2, or an SH, into the drug molecule to produce a more water-soluble compound. The compound now will either be polar enough to be excreted or can undergo phase 2 reactions in addition. In this step, in phase 1, drugs undergo functionalization reactions of oxidation, reduction, or hydrolysis, mainly catalyzed by CYP450, FMOs or MFOs, flavin-containing monooxidases, and epoxide hydrolysis. Phase 2 are termed synthetic, whereby two reactants interact to form a bigger product, conjugation reactions. Conjugation is when you're adding a conjugate group to a drug. So phase 2, synthetic conjugation reactions, conjugate or attach a small polar hydrophilic ionizable endogenous compound, with one exception, the CH3, to the functional groups of phase 1 metabolites or parent compounds which already have a suitable functional group to form water-soluble conjugated products, thereby facilitating drug elimination. Conjugated metabolites have increased molecular weight, improved water solubility, and generally devoid of any pharmacological activity and toxicity. However, of course, we have exceptions to this rule. Phase 2 reactions are generally catalyzed by superfamilies of UGT, SULT, NAT, and MT. Reactions catalyzed by the NAT or N-acetyltransferases are slightly polar. However, those catalyzed by methyltransferases are nonpolar, and this is the exception I've been talking about. As previously discussed, enzymes can either be classified into the MFO isoform, CYP450, mainly CYP3A4 in highest quantities, and other non-isoform like aldehyde dehydrogenases or monoamino oxidases. You can see below phase 1 and phase 2 descriptions showing that, as I've said, CYP3A4 is in highest percentage or highest frequency, and phase 2 reactions mainly are highly categorized by a big portion of UGT glucuronidation reactions. Phase 1 helps introduce a polar functional group and helps prepare for phase 2. So the sequence could be as follows: phase 1 followed by phase 2. But there's always an exception. If the drug has already a functional group that's ready for phase 2 reaction, phase 2 can undergo first of all before phase 1. There's no rule that mandates to have phase 1 before phase 2, like in the case of isoniazid here. We can have phase 2 catalyzed by the NAT enzyme on the hydrazine moiety and allow the formation of phase 2 metabolite N-acetyl isoniazid, followed by phase 1 hydrolysis reaction to form N-acetyl hydrazine moiety and isonicotinic acid. To our opening scenario. Because both risperidone and paroxetine appeared to demonstrate a therapeutic effect for YH, the pharmacist recommended that the dose of risperidone be decreased to 2 mg daily, while continuing paroxetine 20 mg daily. At her next appointment the following month, YH stated that her mood was getting back on an even keel, and her left hand was no longer shaking. Risperidone is an antipsychotic agent that acts through the inhibition of D2 and 5-HT2 receptors. If serum levels exceed those associated with safe therapeutic use, adverse effects associated with excessive D2 receptor antagonism, including tremor, can commonly manifest. Like many psychotropic agents, risperidone, as can be seen in the picture below, is metabolized by CYP2D6. This active metabolite generated is 9-hydroxyrisperidone. Paroxetine, on the other hand, is an antidepressant that works by inhibiting the reuptake of 5-HT. It's also as well metabolized by CYP2D6, yielding an inactive catechol metabolite, thus which can be further oxidized to form an orthoquinone metabolite. Now, why is all of this important? Co-administration of paroxetine and risperidone would result in competition for limited quantities of CYP2D6. As risperidone serum levels increase, the side effects of tremor, dystonia, and akathisia could responsibly be anticipated. Maintaining the two CYP2D6 substrates in YH's mental health drug regimen demands a reduction in the dose of risperidone so that the metabolism-mediated increase in serum concentration keeps blood levels within a therapeutic range and out of the toxic range. So the reduction in dose from 3 to 2 milligrams appears to have accomplished this goal. Most importantly, you can know the rate of drug metabolism, which can help you control the duration and the intensity of action of many drugs by controlling the amount of the drug reaching its target site. Second, you know the products of drug metabolism, being toxic, more active, less active. Third, you can know the competitive and non-competitive interactions during metabolism, and this can help you control DDIs. And fourth, you can know the possible products of drug metabolism, which need to be documented for newly discovered drugs. So drug metabolism influences deactivation, activation, detoxification, and toxicity of the vast majority of drugs. Our main reference was Foy's Principles of Medicinal Chemistry, the 2020 8th edition. And we hope you enjoyed our presentation and we hope everything was clear. You can hit us up in the comments if you have any question, and like and subscribe for more.